All right, well, welcome everyone. Again, I want to first of all thank Laura and Tonya, both for those introductions, but also to add to my regret that we can't meet you in person as we normally do. But, you know, I'm hoping this isn't the new normal, but certainly it's the new normal for this year. But let's take advantage of that. So what I am going to do is give you what I traditionally do is provide a scientific and medical update in person. This year we'll be doing it on Zoom. So without further ado, I will try and step through these slides. Let me see. There we are. So as many of you know, I do wear, unfortunately, many hats. So I'm speaking to you today in my capacity as medical advisor to the BBS UK Society, both as a clinician and a scientist. Just a couple of declarations of interest. I will be talking about the first one a little bit tomorrow. So, as Tonya has already introduced, 10 years ago, and this year we're celebrating 10 years of the highly specialized service throughout the UK for Bardet-Biedl syndrome. It's been an incredibly fruitful journey, but it's been one that is, I think, equally welcomed by our patient community and has really led to quite a lot of not only interest, but many accolades from the NHS itself. And there are many other services that have been built on this particular model. And what I'm showing you here is really 6 elements that comprise that model. And essentially it's made up of 4 different hospitals, 2 in London, 2 in Birmingham, where we see children who then transition when they get to 17 or 18 to their respective adult hospital. But pivotal to this particular service is the work of the charity BBS UK and the officers who make up the service, who provide unparalleled support, which we could not provide as clinicians within that service. And what we're able to do, therefore, is to provide lifelong care for these patients, which normally would be lost to follow-up, probably around the age of 16 or 17. So we have a lot of information, a lot of data, and we know all of these patients absolutely, well, personally, but in an absolutely holistic way, if you like. Another component of this particular service, the 6th piece, the 6th linchpin, is the DNA lab. And that provides a national reference service for the whole country. And this has been ably led since day one by our colleague, Dr. Beth Hoskins, through the Great Ormond Street Hospital Genetics Center, whereby we've been able to offer a next generation Bardet-Biedl syndrome gene panel, which comprises 21 genes. And we've now, this is the data from 2020 I'm showing you here. So it's already increased by about 50. So we're talking about 900 tests that have been completed to date. And we've been able to confirm diagnosis of BBS. That's in patients who fulfill the genetics criteria, as well as having this supportive molecular diagnosis in around 80% of the families that have come to the particular service. We've done 224 carrier tests, and we undertake a number of predictive testing, particularly prenatal testing, for couples who are particularly worried about whether their pregnancy may or may not be affected. Also, we're able to define a whole host of different mutations as well, around 200 mutations now across the board in all of those genes. And importantly, we also know that around 22% of patients that we see do not have a mutation, and there can be many reasons for that. And in general, what we see is around a steady state of about 33 new referrals coming to the service every year. We can look at this in a different way, if you like, now, by a pie chart. And the pie chart, let me just get the pointer up here. I don't know, can you see a pointer there? There we are. And essentially what we can see is that the majority of these are represented by mutations in the BBS1 gene, followed closely by BBS10, and then here's the 22% unknown. And we can see common mutations that are present amongst this group as well. I won't dwell on this, but we can also break all of this information down by mutation type, which in the future may become incredibly valuable when different therapies come online. On the back of the clinic and taking advantage of the pandemic period and the various lockdowns, I finally got around to building our BBS Patient Registry with the help of several colleagues who I'll shout out in just a moment. But why would we want to build a patient registry? Well, we want to be able to constantly provide clinical improvements to the service, both in terms of care and specific service implementations, if you like, undertake audit, undertake research, and also to support clinical trials as they become available. And we want to be able to provide this to not only the other BBS centers so they can see what's going on as a whole, but also other colleagues within the NHS itself, as well as clinical researchers and trial centers going forward. We've built a robust governance structure around this, including a registry committee that includes lay people, such as those from BBS UK and members of NHS England itself. And we have built this particular database called Redcap with the help of the registry team, who have stepped up to the mark during the pandemic and worked incredibly hard to build this out. So I want give a big shout out to both Ian Kemp and Theodora Diakova, who helped to design and build all of the field.
Through it together step by step.
Hello?
Right. Put me on loudspeaker.
Yeah. They've been able to help us build out this database going forward. I just want to give you an overview of what this Redcap looks like. It's a database that has been devised as a secure web application. It is there really for managing both databases and online surveys. But what we've been able to do is to build out 12 different forms, and I'll show you those in just a second. Most of these forms are repeating, so we repeat one for each new visit. So far we have over 300 records uploaded, but within the next couple of months we're expecting to double that as we bring on the other centres around the UK. And what you can see here is those particular 12 instruments or fields, if you like, listed in the columns here. They include all the general demographics around individuals, whether they fulfill the major or minor diagnostic criteria, what their height, weight, BMI might be, what their blood pressure might be, what their genetic scores are, what the endocrine, the kidney, the blood results all look like. So we can dig a little bit deeper, say, into the endocrine field, and we get all of these various drop downs that have been carefully designed, because it's important to just have drop downs and not too many free text type fields. And we can capture, for example, the diabetes information here for this particular individual, and the same applies for lots of these other patients other fields going forward. And then what can we do with this information? Well, we can do a number of things. We can understand better what the patient journey looks like. we can also understand the natural history of Bardet-Biedl syndrome. For many years, we think we've understood the natural history, but it's only when you start looking at a great detail in a registry such as this, you begin to really understand what is the commonality around the onset of various features. So for example, we can see 84% of these patients are born with extra fingers and extra toes. Within the next couple of years, we're going to start to see that around 90% of patients, 88% specifically, will start to show signs of developmental delay or behavioral disturbances. And then we start to see the onset of weight gain, the obesity. And then it's about 4 years of age that we're starting to see the first signs of visual impairment in the form of night blindness and so on. And then we can go on, we can see about almost 40% of patients will have renal problems, 26% will have diabetes, and 47% will have hypertension. And then this is going to be incredibly valuable information as we move forward, as we start thinking not about just about service improvement, but also about treatment in general. And so we can take and dig deep into the database. We can start looking at information, and what we've done here, for example, is to analyze the body mass index, the weight, and you can quite clearly see when we place that black line on the graph here, which is the population norm for the UK, that virtually all patients sit well above that line, and it increases with age, as one might expect. Okay, let's get into the research. What I like to do is sort of go back over the past year since we last met. Well, actually, it's been a couple of years now, and just have a look at some of the key research that has either come out of our own labs or come out of other labs around the world that we think are having a major impact on our understanding of Bardet-Biedl syndrome. And what I can say is that there is this inexorable interest in Bardet-Biedl syndrome as an entity, and we can see clearly here since 1946 the steady number of publications that there has been. But around 17 years ago, this suddenly jumped. And that probably coincides with when we found the first gene, as Tonya mentioned. And since then, it's been increasing steadily. So this year already, there have been about 54 publications to date. So I expect that to be, well into the over 100 by the end of the year for sure. And what we're finding is that there's an increasing number of case reports from all around the world, and just some of those are listed here at the bottom. But one of the questions that have been perplexing us for many, many years is, what is the cause of obesity as one of the key cardinal features of Bardet-Biedl syndrome? And I think one of the things that has really come to the fore in recent years, and particularly in this last year, has been the role of the cilium. So I know a lot of you in the audience will be familiar with cilia, but others may not. So just a quick primer here. So primary cilia are short membrane protrusions that emanate from the surface of virtually all the cells in our body. They function, I guess, in the most simple manner as a cellular antenna, and they receive information about the environment, and they locally transduce that information into a cellular response. And various signaling pathways can exert their function through the primary cilium, and they do this through a number of pathways that include things like, funky names like sonic hedgehog, Wnt signaling, and something really important called the G protein coupled signaling, which uses a secondary signaling molecule called CAMP, which we'll come back to in a moment. But it's notably the function of primary cilia that really depends on an array of proteins that maintain the structure and the signaling dynamics of the cilium itself. And what we now know is that mutations that encode a number of these defective ciliary proteins underpin ciliopathies in general, and not least Bardet-Biedl syndrome. And it also helps to explain why, or sort of newfound understanding, if you like, why there is retinal degeneration and the visual impairment, cystic kidneys, even the presence of extra fingers and extra toes. And now we're beginning to better understand the role of cilia in obesity as well. As I mentioned a little bit earlier on, there are 21 or 22 genes now associated with BBS, but 8 of these genes encode for proteins that form a protein complex called the BBSome. The BBSome itself is absolutely vital for the function of the cilium, but it doesn't directly contribute to ciliary formation, but rather controls the sort of the dynamic localization of other proteins in that cilium. And in particular, these G protein coupled receptors, or GPCRs, some of them are called melanin concentrating hormone receptor, for example. Another one will be called neuropeptide Y receptor, and another one might be somatostatin receptor, and so forth. But what's really important is that each of these proteins are signaling to the inside of the cell through the secondary messenger protein called CAMP. And what we're now beginning to understand is that it is through this particular process throughout the body, but particularly now in neurons in the brain, that these proteins are having a major impact. So what is obesity? Well, it's essentially in its simplest form, an imbalance in terms of energy intake, right? So, you know, the intake has to match the output. And quite clearly that imbalance is prevalent within patients with BBS, but also with mouse models. So we and others have made several mouse models of BBS, and they recapitulate that level of obesity. And what we now know also through the work of others is that specific deletion of BBS proteins, such as BBS1, in key neurons in the brain, such as what's called POMC neurons or agouti-related protein neurons, which reside in a part of the brain called the hypothalamus, in particular cells called the arcuate nucleus. This forms the energy control center in the brain. And we know that dysfunction of BBS1 proteins, or absence of BBS1 proteins, now leads to increase in food intake and thereby increased weight gain. And what we're beginning to see now is getting a better understanding of what is going on. But up until now, there has not been a really good human model of disease to be able to understand what's going on. So before I go on to the study that I want to share with you today, I want to talk about these things called induced pluripotent stem cells. And many of you or patients who have come to our clinics in the last couple of years have been asked to take part very kindly in a study where we took skin biopsies. And we turned the skin cells, which are what we call differentiated, back into less differentiated cells, stem cells or progenitor cells, if you like, called IPSCs. And in previous conferences, I've shown you that we have in the lab been able to create some kind of mini eyeballs in a dish, as you can see here on the left-hand side. And they form all the layers of the retina, including early stage photoreceptors. And essentially what this paper, which has just came out earlier this year, shows from Rudy Leibel's lab at Columbia, was that they were able to generate neurons, and specifically neurons in the hypothalamus that are important for, the type that are important for appetite suppression, for example. And they were able to take these from these IPS cells, which were originally derived from those skin biopsies. And then they wanted to look at what the effect of deficient BBS1 or BBS10 was. And so they do this in a very complex way, which I won't go into today, but the take-home message is that what they essentially found was a number of morphological defects in these particular cells, in these nerve cells in the brain, in the appetite center in particular. They saw that there was decreased insulin signaling. They saw that there was shortening of the neurite lengths, so these little protrusions that are coming out of the nerve cells and the neurons. And there was a decrease in the number of these as well. But there was also an increase in the cilia length of these particular of these as well. And we also saw in this study a decrease in key signaling, the feedback mechanism that tells us when we're full, for example, leptin signaling and insulin signaling. So in addition to this sort of these changes in the morphology of the cells, what was also found was that there was key ciliary signaling deficiencies as well. Sonic hedgehog, Wnt signaling were all deficient as well. So for the first time, we now have a really robust in vitro model in which we can test BBS mutations and other ciliopathies that are associated with obesity, for example. And but what they went on to do is also have a look at what's called gene expression profiling in these hypothalamic neurons that they'd made. And then they were able to show that, confirm, should I say, that insulin leptin signaling was all deficient, and that this was all happening through this module of the CAMP signaling pathway. So now we have a really good model, as I say, to be able to move on and better understand the cause of obesity. But what they were also able to show in this particular study is that restitution or expressing the wild type BBS protein was able to correct these particular problems completely. And so for us, as we'll talk about tomorrow, this also leads to the fact that, as we are attempting to do, provide gene therapy, in other words, to replace or restore, replenish that particular missing gene within those particular cells. And then finally, other studies as well have determined that cilia, in general, primary cilia, are really quite important now for regulating weight in general in all of us. And that probably at the heart of this really is the BBSome. So what we're expecting to be able to see over the next few years is the role of the BBSome its role in causing generalized obesity, and potentially therefore therapies that might go with it. So then just a couple of other studies I wanted to share with you, just to highlight some interesting, what I felt was some interesting components of Bardet-Biedl syndrome. And this particular study from Benades, also that came out in October, end of last year, published in February this year, essentially showed that BBS proteins not only are present in neuronal cilia, but also in other parts of the neuronal cells as well. I mean, in other words, in particular cells in the brain that are myelinated. So myelin sheaths that sit around axons that come out of nerve cells, these incredibly long cells that pass through the body. The myelin acts as a kind of capacitors, if you like. They act to make that process of signaling to, say, your big toe, for example, very, very fast. And we know from various diseases like multiple sclerosis, where myelin breaks down, that this process does not work very well. And what we're now seeing in this particular study is that BBS proteins, in particular in this case BBS4, is now found in myelinated cells. and particularly in the periphery of myelin sheaths. So the question really is there a role, therefore, not only for BBS and BBSome in myelin formation, but also is there a role in BBS? Because we know a lot of patients do have peripheral nerve problems, and that will be an interesting issue to take up in future years. And then the final study I really want to just share with you is a study we had the good fortune to work with Andrei Stepanek's group from Charles University in Prague a year ago. And what we were able to do in this particular study is, first of all, to determine that there were immunological defects associated with BBS. And this actually determined that there were a number of autoimmune features that seem to be more common in patients with BBS than people who do not have BBS. So we reviewed the CRIBS and the UK BBS registry, and we saw that there was an increase in the amount of rheumatoid arthritis, type 1 diabetes, hypothyroidism, and a whole host of other conditions that are considered to be autoimmune. And then Andrei's group was able to show, using a particular mouse knockout model, again of BBS4, that there were abnormalities in the B cell group of immune cells, not the T cells, but predominantly the B cells. Even though they were able to show these abnormalities, we don't quite know what the link is, so there's quite a lot of extra work to be done here, but quite clearly this starts to shed new light on the potential role of primary cilia in controlling the immune function, not only in BBS, but in disease in general. So at this point, I just want to summarize really and just say, we now have a better understanding of the cause of obesity in BBS, and that we can now start to see a potential for several new therapies going forward. We've seen that BBS proteins are now found in a number of nerve cells that's sitting outside the cilia, but in other parts of the cell, like in the myelin sheath, for example. And we're also now asking ourselves the question, are BBS proteins important for immunity? So I'm going to leave it there. Thanks for your attention. And we'll come back to take any questions very shortly in the question and answer session. Thank you.